The genome.ucsc.edu homepage links to many tools and resources in addition to the Genome Browser. In this video, we will be focusing on the SARS-CoV-2 Genome Browser, so let's navigate to it. Here on the Genome Browser, you are seeing the entire span of the SARS-CoV-2 reference genome identified by its RefSeq accession along with the default data tracks. The default view shows full-length genes from NCBI and UniProt. For example, ORF1AB, which is cleaved into a whole bunch of different protein products. You can see when I hover over a gene, there's a hover title describing the function of that gene. The UniProt highlighted regions of interest track shows the location of the receptor binding domain and other interesting features. The next track, UniProt domains, is in dense mode, but we can expand it to learn more by clicking it. Now we can see well-characterized building blocks of the proteins for SARS-CoV-2. We can condense the track again by clicking on the title. Next, we have UniProt amino acid mutations. As you can see, most of the annotated variants are in the spike protein region. Expanding the track lets us hover over items to see information curated by UniProt. For example, spike mutation D614G enhances binding with the ACE2 receptor. It's pretty crowded there, so we can zoom in by holding down the Shift key and clicking and dragging to select a region. There's also an option to add a highlight for a region instead of zooming in. The Bloom Lab Serum Average track shows how strongly the binding of antibodies from patients to mutated versions of the spike protein was affected by mutations at each position in the spike protein's receptor binding domain. Since this track shows scores instead of features, expanding it shows a bar graph instead of grayscale shades. If you click on the mini button to the left of the track, then you can add several related datasets to the view. Our Variants of Concern track shows the protein-changing mutations found in each variant. Clicking on the mini button to the left of this track lets you add other variants of interest and/or nucleotide mutations. These tracks are just what appear by default, but if you scroll down, we have many additional categories of datasets that you can add. If you are involved in sequencing SARS-CoV-2 genomes, then you may be interested in our primer tracks. Let's add several of these to the view by changing them from hide to dense mode and clicking the refresh button. Here we can see that the Omicron variant of concern has multiple mutations in some primer binding regions. Despite having several UniProt tracks displayed by default, there is quite a bit more to go into the details and features of protein domains, protease cleavage, glycosylation, phosphorylation, and other protein mechanics. On the other hand, if you're looking at a mutation in the spike protein, maybe the tracks in the immunology section may be of interest to you. You can click on any track's title to see its options and documentation. We also have RNA expression data, structure data, and even comparative genomics data to show which parts of the SARS-CoV-2 genome are conserved across many similar coronaviruses. For example, phyloCSF shows codon substitution frequencies in 3 frames, focusing on protein coding conservation across 44 sarbecovirus genomes. When you zoom out to look at the whole genome, it gives a pretty good signal for where the genes are within the genome. We have various ways to look at mutations, such as the regularly updated NextStrain build. NextStrain selects a subset of several thousand out of the more than 6 million sequences in GISAID that is selected to focus on the most recent sequences with enough older sequences to reconstruct a phylogenetic tree. NextStrain designates clades, which are named by year and letter, aiming for a broader categorization of variants of SARS-CoV-2 than the fine-grained Pango lineage system. Here are the mutations in the several thousand sequences that NextStrain downsampled most recently. Mutations are colored red if they change the protein, green if synonymous, and black if outside the coding regions. The phylogenetic tree is on the left, and at this point in the pandemic, the virus has diverged enough to make this tree pretty deep. We can make more space for it by clicking on the Configure button and increasing the label area width on the left of the page. There are other settings you may wish to experiment with on the Configure page. Even with a drastically widened label area, you can see that the Delta branch just keeps going and going. The next video will show how you can upload your own data to view alongside all the tracks in the Genome Browser.